Alrighty, as the board says, we're going to be talking about cholesterol anabolism. There is no cholesterol catabolism. Our body doesn't break down cholesterol. It's going to use any kind of cholesterol it has in the body. Your body's going to use about 50% of cholesterol from your diet, and it's going to make about 50% of its own cholesterol. Obviously, those numbers can vary depending on your diet. So let's jump into it a little bit right here. Cholesterol is this wonderful clusterfuck. Yeah. So what we have on cholesterol is we have a polar group and then we have a large non-polar chunky. So what cholesterol does as it's in plasma membranes is it reduces the fluidity of the plasma membrane. It would be offset by your unsaturated fatty acids. Cholesterol is defined as an isoprenoid, which means it's made up out of isoprene subunits. An isoprene subunit is that guy right there. This is termed an isoprene. Isoprene. Why? I honestly don't know. It's just there. Isoprene. Cholesterol, good. Some of the things that cholesterol is used for, aside from just membrane fluidity, it's also the basis of bile, as well as biliary cholesterol, bile salts. It's also used in the synthesis of vitamin D. Cholesterol in your skin will be split by UV lights into vitamin D. In addition to that, it's also the basis for many steroid hormones. These are things like testosterone, aldosterone, those wonderful hormones that we've learned about very minimally as of yet. So let's just jump into it here. We know that cholesterol anabolism is another type of lipid anabolism. All lipid anabolism uses NADPH as an electron carrier. Again, we have two sources for NADPH. That is the pentose phosphate pathway in the case of carbohydrates, and that is malic enzyme in the case of amino acids. When we're making cholesterol, we have an excess amount of energy. If that excess amount of energy is from carbohydrates, we have glucose 6-phosphate to run the pentose phosphate pathway. We'll get our NADPH from there. If we do not have carbohydrates as the source of our excess energy, our body will not be making glucose 6-phosphate to run the pentose phosphate pathway. It'll break those amino acids down and then use the malic enzyme to get your NADPH. So cholesterol anabolism, lipid anabolism using NADPH. It has 4 separate stages, and we're going to run through all 4 of them right now. The first stage, stage 1 of cholesterol synthesis, is essentially just the first 2 steps. The first 2 steps of ketone anabolism, followed by a different reaction that occurs on the smooth endoplasmic reticulum. So stage 1, we're going to start off with 2-acetyl-CoA. If you haven't already, watch the video on acetyl-CoA transport. That's super integral to this. That involves the malic enzyme, which is going to be producing the NADP. So we've got an excess amount of acetyl-CoA, so we're going to take these and we're going to bind them together. Bam, right like that, into acetoacetyl-CoA. This is via the actions of the enzyme thiolase, or acyl-CoA acetyltransferase. If you want to use the full name, acetyltransferase. If we look at these 2 compounds right here, we have 2 acetyl-CoAs, which means we have 2 CoA groups. Right down here we have 1 acetoacetyl-CoA. There's only 1 CoA group, so we are going to lose a coenzyme ASH in this process. The hydrolysis of this high energy bond is used to give the energy to create the bond between the acetyl groups there. The next step in the pathway is going to be essentially the same thing. You're going to take another acetyl-CoA and you're going to tack it onto the compound. This is via a slightly different enzyme. and that enzyme is different because of the compound here. The compound that's synthesized is called beta-hydroxy, beta-methyl gluteryl CoA. Holy, that's a mouthful. However, it's also known as HMG CoA. Again, we have 2 coenzyme A's up here, only 1 coenzyme A down there, so we're going to lose a coenzyme ASH. The enzyme that does this is wonderful because it's making beta-hydroxy beta-methylgluteryl CoA, or HMG CoA. It's actually called HMG CoA synthetase. Synthetase or synthase? I think just synthase. Synthase. HMG CoA synthase. These are, again, the first 3 steps in ketone anabolism. In ketone anabolism, we have an excess amount of acetyl-CoA from some kind of energy source that we're trying to use in some manner other than the TCA cycle, because the TCA cycle enzymes are all taken up by the acetyl-CoA that is already there. So, moving on from this, these are exactly analogous with ketone anabolism. The next step is where it's going to divide from ketone anabolism and cholesterol synthesis. So, and this is still stage 1. This is going to be the last step of stage 1. We've got our HMG CoA that was made by acetoacetyl-CoA and acetyl-CoA, as well by the enzyme HMG CoA synthase. We are now going to alter this structure slightly. What we're going to do is we're going to take an NADPH plus H plus, and we are going to dehydrogenate this compound to form something called mevalonate. This is the last step in stage 1. It is also the rate limiting step. This is going to be the regulated step. The enzyme that we're using here is HMG CoA reductase. And HMG CoA reductase is found as an integral membrane protein on the smooth endoplasmic reticulum. So it crosses the entirety of the smooth endoplasmic reticulum plasma membrane, and that is where it would be found. Again, we have a coenzyme A up there. We don't have a coenzyme A on mevalonate. So what this is doing is we're releasing that coenzyme ASH. Now, how many carbons is in mevalonate? Well, we know that we had 2 acetyl-CoA to start with, and we added in 1 more acetyl-CoA to make HMG CoA. Each acetyl-CoA has 2 carbons, so mevalonate is a C6 compound. This is